Okay, let's talk about methionine for a minute because that is the initiator. As a matter of fact, when we said that our messenger RNA went to a small subunit, it actually the very first codon had to be AUG because that coded for the amino acid methionine, and when methionine hooked up with that, it caused the large or heavy subunit to come together to form the whole ribosome, and then the ribosome can go read the rest of the messenger RNA. So it's important to understand that the very first one, so if this were the very first one right there, it would have to be methionine. So looking back at methionine, we know then our DNA sequence, our coding strand would have been ATG, our template strand, which we then read once our DNA was unzipped, was TAC. So therefore, our codon that we formed for this segment, A would be complementary to thymine, uracil would be complementary to adenine, and guanine would be complementary to cytosine. Then when the messenger RNA actually left and went to the ribosome, then the anticodon, the transfer RNA, which was actually bringing the methionine, its anticodon had to be complementary to the codon. So the anticodon for methionine is always uracil, adenine, cytosine, because it's always complementary to the adenine, uracil, guanine that came on the codon. Now, I hope that's not too confusing, but anyway, that's an important point. And the other videos I asked you to watch also brought this out. Okay, so again, looking at this, all of these, whether it's a complementary triplet, a codon, or an anticodon, all of this row codes for that amino acid. All of this row codes for this amino acid. AAA will never code for leucine. AAU will never code for phenylalanine. They're very specific for what they code for, so our information tells us the order. Okay, let's look at this actual codon chart and see how the codons code for the amino acids. Now notice that I've got my 4 bases here, uracil, cytosine, adenine, and guanine. My 4 bases going across the top, uracil, cytosine, adenine, and guanine, and my 4 bases going across the side. So that's basically 4 to the power of 3, which means then that I basically have 64 different blocks here, because 4 times 4 is 16, 16 times 4 is 64. All right, now notice that what I have in these blocks, I have both the codon, the 3 base sequences, all these have and then I have the name of the amino acid that they code for. Now I actually showed you guys the 20 different amino acids earlier, not that you had to memorize them or anything, but you wanted to see what they actually look like from their structure and what their properties were. But what we're looking at now is, notice that if I actually look at the different names, I would see that I have 20 different amino acids spread throughout 64 blocks, with a couple of different blocks here that are unique to some other things we'll talk about. So for instance, let's say that my codon was CCA. Well, notice CCA, cytosine, adenine, only codes for proline. It doesn't code for glutamate. It doesn't code for histidine, arginine, or any of the other 20 amino acids. But I also want you to notice I actually have 4 different codons that will give me proline. Okay, so whether it's CCU, CCC, CCA, CCG, they will all give me proline, but CCC will only give me proline. It will never give me any of the others. Here we see the same thing here. CUU only gives me leucine. It will never give me any of the other 20 amino acids. Here I've got CUC, which will also give me leucine. CUA will give me leucine. CUG will give me leucine. But they will never give me any of the other amino acids. Let's look at this example here real quick. We've got CCG. That is my codon. That it happens to be my second codon, CC, and then we said that was G. So let's look and see what amino acid would that code for. So I go here, it was CCG, so I start over here, C. So I know it's somewhere in here because these are all C's. Go to my next column, C. So I know CC is somewhere through here, and then finally my last column. So CCG would be proline, so I know that that codon would only code for proline. And I know that what would bring the proline to the ribosome would be the transfer RNA with this anticodon. So I know the anticodon for CCG then would be GGC. Okay, so let's look at another one. Okay, let's look at this next codon, which was AGC, and let's see what would that code for. AGC. So what we would do, we go to A first, knowing it's somewhere in here. We go to G next, knowing it's somewhere through here, and AGC. So that means the next codon would have coded for serine. And notice it doesn't code for anything else. Now there may be other things that code for serine, but that specific codon gave me serine. So what would be the anticodon for that? Well, since it's complementary, we know that what's complementary to A would be U, what's complementary to G would be C, what's complementary to C would be G. So UCG would actually be my anticodon that would literally have brought serine. But keep in mind, this is a codon chart. This will be the chart that you will see on the test. This will be the chart that you will have to use to determine the amino acids. So I've given you some practice things, so let me start you out and show you how to do this. And then if you have trouble, you can email me or let me know, and I'll try to give further instruction or put the answers down. But first thing here, what we've got, I've given you the template strand. I've had to give you something from this. So I literally gave you half of the DNA, the template strand. So I gave you these template strands actually have 3 base sequences that we actually call these triplets. All right, so I gave you TAC. What I'm asking you then is what part of the coding strand is complementary to that TAC? In other words, what is the complementary triplet? So first thing we say, well, what's complementary to T? And of course, the answer would be A. What's complementary to A? Well, the answer would be T. What's complementary to C? Well, the answer would be G. And again, remember, this is DNA, so we don't have uracil in DNA. We have adenine, guanine, cytosine, and thymine. But once we start forming RNA, we have to be careful because we have uracil instead of thymine. All right, let's do one more here just to make sure we've got this part. We'll do 2 just to give you an example. So if I said, well, what's complementary to G here? Hopefully you would say, well, that's easy. That would be cytosine. And since cytosine's in both, we can put it. And if cytosine's complementary to that G, then that means cytosine's complementary to the next guanine, or G. And then if I said, well, what's complementary to C? Then the next thing, of course, would be guanine. Okay, so here I've got my coding strand and my template strand. But remember, when our enzyme comes through and unzips it, it's not going to read the coding strand, it's going to read the template strand. So if we're going to form our messenger RNA, then we need to read this strand. Okay, so first of all, let's say, well, what is complementary to T? Well, what's complementary to T is adenine, okay? And so we say, well, does RNA have adenine? And yes, it does. So here we put our A. Next then is, what is complementary to adenine? Well, thymine is, but RNA doesn't have thymine, so what else is complementary to adenine would actually be uracil. So now, since we're making messenger RNA, then the answer would be uracil. And then we say, well, what's complementary to cytosine? And we say, well, it's guanine. Does RNA have guanine? It does, so we can put it. Okay, so this, and this kind of all line up with each other. So the next thing then is let's read our guanine, and cytosine and see what our messenger RNA would be. So what's complementary to guanine? Well, it's cytosine. And does RNA have cytosine? Yes, it does. What's complementary to guanine? Cytosine. And what's complementary to cytosine? Well, guanine. So notice that my messenger RNA looks a lot like my coding strand, but this is RNA because in the nucleotide, remember, it's got ribose sugar instead of deoxyribose sugar. But as far as my 3 base sequences, which now these are called codons, so with my codons, I've got AUG, then CCG. Now, once I build this whole messenger RNA and it actually leaves the nucleus and goes out to the ribosome, then this AUG goes to the ribosome first, and now transfer RNA has to come try to read it. So it also has to be complementary. So which nucleotide is complementary to A? And since it's RNA, we know it has to be U. So we'll put a U there. Okay, what's complementary to uracil? Well, adenine. And what's complementary to guanine? Cytosine. Okay, well, let's read then. We just read our first codon with our first anticodon. Now let's read our second codon and determine what our next anticodon would have to be. Well, if that's cytosine, then obviously it would have to be guanine and cytosine, guanine and guanine cytosine, because RNA has all of those. All right, now the next thing then would be to determine from our chart what would the actual amino acids be. Okay, so we've got AUG and CCG because and the reason why I'm using this line is because the chart that I'm giving you is actually a codon chart. Okay, so looking back at this, then I know I need to read this line because this is where my codons are. AUG, if I look for AUG, notice A means it's somewhere in here, U means it's not just in here, but it's in this area, and then G means it's on this line. So notice this says start methionine. So AUG would literally be, and it says using information from the previous page, give the amino acid sequence. So the very first thing here would be my start codon methionine. Okay, now my next codon was CCG. So going back to this, C means somewhere in here, CC means it's somewhere in here, and then CCG means proline. So my next amino acid in this case would be proline. Now, what I want you to do is go ahead and try to work through the rest of these sequences that I gave you from the worksheet that I posted. See if you can come up with the same thing, and then I will post the actual answers and see if you were able to come up with the right answers. And if you have questions, make sure that you email me on this, because you will have to do this on the test. So I will give you very similar problems. Of course, I will basically give a different sequence of events, so you have to come up with different numbers. But remember that because we always have to start out with our start codon, then that means that the first messenger RNA always has to be AUG, and the first transfer RNA always has to be UAC, because that's the only thing that will initiate the transcription and the coming together of the heavy and light ribosomal subunit. After that, it could be a variety. But let me talk about one more thing before this finishes, is that notice where I only have one start codon, I literally have 3 stop codons. So I have 3 different codons that could literally tell me to stop building the developing polypeptide. And so my last answer here has to be one of these 3 to tell me to stop. So even in looking at this exercise, notice ATT. Well, let's see if that's one of our 3 stop codons. So ATT would have given me then UAA from my messenger RNA, which is the codon strand. So let's see if UAA would actually give me a stop codon, and we can see UAA. There it is, stop. So that would have to be the last. So if you're not starting out with methionine and you're not ending with a stop codon, that means what you probably did was you probably read the wrong line. We're using the wrong base sequence for the chart. So try not to do that. Okay.